Good morning and welcome to day two on the synapse, the neuron and neurotransmission part of the advanced series. And today we're going to be talking about the very fundamentals about how information is gathered, processed, and transmitted from one neuron to the next. And the most important piece of that relevant to addiction is what's called the synapse. And so there are three parts of the synapse. And the one, just to begin, the synapse is at the end of the axon that we talked about yesterday. The cable that transmits electrical wave action potential, and it goes to the end of the axon to what's called the presynaptic terminal. And between the presynaptic terminal, there is a water-filled space that is electrochemically neutral, and that's where the neurotransmitters turn be released into to then interact with the postsynaptic terminal on the next neuron. That's where the next neuron begins on the other side of the synaptic cleft. And these happen, for the most part, on dendrites, on the dendrite bouton, the little knobby part, which is essentially off the tree branches of the dendrites, which act as postsynaptic terminals where receptors reside. And so axon terminals are referred to as presynaptic terminals. They mean essentially the exact same thing. And so they're located at the very end of an axon. And whereas in the initial Tedson-Chapel series on the neuron, we only spoke about neurons having one synaptic terminal. Now, there is only one axon that comes out of every neuron, but the axon itself at the end will divide into many, many terminals. They're all relaying the same information, and they are the presynaptic terminals as part of the three-piece synapse. And so the terminal regions, they often appear as bulbous arrangements that come off of branches of the main dendrite. And the branches are called spines, just for interest's sake. And the axon terminals are really incredibly well organized structures to be able to survive metabolically through resting state homeostasis, and to be able to respond as needed to stimuli. Within the presynaptic terminals, there's, just like within the cell body, there are clusters of what are called organelles, meaning little component pieces, including mitochondria, which provide the energy for the terminal to function. And as well, there is some pretty complex biosynthetics, meaning proteins that are endogenous made within the neuron to do such things as neurotransmitter transport, ion transport. And so the energetic and biosynthetic support all exists within the actual axon terminal to be able to ready synaptic function at all times. And so here's an electron micrograph within the square. The square is surrounding a part of a presynaptic terminal. And within that square, you can see round circles. Those are the vesicles containing molecules such as dopamine. And they organize themselves around the presynaptic membrane, which is the darker area as I'm following along with the cursor. That's the presynaptic membrane. And the space on the other side between the postsynaptic terminal, this is actually the synaptic cleft, that inert water-filled space, and it is approximately 50 nanometers in depth or in width, fairly consistently across its entirety. So these structures appear, the boutons are these little knobbies at the end of axons that are presynaptic terminals. And they serve both for storage of neurotransmitters, release, and maintenance of the amount within the terminal itself. There's numerous synaptic vesicles within each presynaptic terminal with specific different types of neurotransmitters that are organized actually into distinct pools, say a dopamine pool, say a GABA pool, so that on stimulation, the excitatory or inhibitory neurotransmitters are released depending upon the input. And so, how does this happen? There's always, just so you know, and this is relevant, there's always a general circulation of neurotransmitters, even at rest, in your sleep, all day long. We've talked a lot about what I refer to as the low dopamine state in early recovery. And that, what I'm talking about is, in general, there is a certain amount of dopamine that is released and flows into the synaptic cleft, activating the neuron on the other side of the synapse and recirculated back into the presynaptic terminal. That's just going on all the time. And so when I talk about low dopamine states and talking about the motivational neurons, the reward neurons, there's less of that circulation going on, whether it's actually less dopamine molecules available or resistance in the circuit or both. That remains a question to be discovered. So now we're going to talk about what actually happens when, and I'll just show you here the representation of an electrical charge, an action potential, electrical current that flows down the axon. How does that translate that an electrical current actually magically spills the dopamine vesicles into the synapse? Well, what actually happens is that the electrical current creates an elevated calcium ion concentration that activates calcium sensor proteins, snare and snap proteins, they're called, which actually, on the basis of electric charge, draw the vesicles, the neurotransmitter vesicles, say the dopamine vesicles, to the synaptic membrane, where they fuse together, they stick together, and then release. This is called, that whole process, if we go back to the basics of the first month here, we call that neurotransmitter transporters. Just to sort of simplify the whole thing about what a transporter actually is, we just put a name to it. This happens. Transporters take dopamine to the cell membrane and release. Well, here we're doing deep dive, and it's actually the way that it's happening is that positive calcium ions are activated with electrical charge, and that then attaches to the vesicle and electrically draws them towards the electrically neutral synapse. So in essence, this is what short form is neurotransmitter transport. Interestingly, and relevant to our discussions, crystal meth, methamphetamine, works its way into speeding up the snare and snap protein actions, thereby dumping more and more dopamine into the synapse, as well as working on transporter or neurotransmitter reuptake. Remember, cocaine blocks reuptake, and crystal meth blocks reuptake plus enhances transport, which is why dopamine levels with crystal meth can hit 1400 versus cocaine, which is about 700 maximum. And so here, this diagram illustrates the where the arrow is, illustrates, again, an electron micrograph of this, shows the actual neurotransmitter vesicle fusing itself to the cell membrane here and really actually releasing contents into the synapse, such as here. These are free-floating active neurotransmitters. They can be free-floating active dopamine molecules that can then activate receptors on the other side. And so the fancy word for this is called exocytosis, meaning exo meaning out of, cytosis is cell. So out of the cell, release into the synaptic cleft. And so as this cycle continues, the vesicles themselves are retrieved and actually recycled, and through reuptake transporters refill with neurotransmitters to be ready for the next release. And that's called endocytosis, meaning endo into cytosis cell. And so what we have is essentially a neurotransmitter recycling plant. The neurotransmitters are released, touch, activate receptors, and then are recycled back into the cell, the presynaptic terminal. And as I said, at rest, this is a continuous process so that there's constant circulation. You're not depleting dopamine, not overstimulating, just essentially homeostasis, keeping the neuron alive and stable. And so, again, building on what I spoke to 2 slides ago, we have an illustration of a presynaptic terminal at rest, which is at the top left, showing vesicles within just floating around the presynaptic terminal. And at the same time, they're organized, meaning they're pooled and localized so that the larger, closer to the synaptic membranes are going to be the ones activated. And so the at rest is then stimulated, meaning an electrical current is introduced, meaning these axon fires, and we see at the bottom what happens with the stimulated, that the vesicles then move to the membrane, fuse to it, and you can see an indication that there is release of neurotransmitter molecules, dopamine, say, into the synaptic cleft. And so rest close to the membrane, stimulated, fuse to the membrane and release particles. Now, it's good to know that at rest, you can think of the neurotransmitter vesicles as in a docking position. They're docked at active zones, meaning ready to open. And the process here, and this matters, when one really drills down on the pharmacology of addictive substances, what happens in terms of this stimulation through the electrical current, the action potential down the axon, and the calcium molecules actually open up gates. It's called their gates. And those are places in the membrane that open. And so with, say, crystal meth, the action most likely of crystal meth speeding up neurotransmitter transport is it opens up those G protein channels, also called voltage gated. Voltage meaning there's got to be an electrical charge. They act a voltage gated channel is stimulated electrically. No electricity, voltage is zero across the gate. Add voltage, add a charge, gate opens. That's likely where crystal meth actually works. And so fascinating how this really comes together. You can see on this diagram at D, there are the three stages of signal transmission. And the first stage, labeled 1 here, and 1 on the graph. If you look at D, if you look at the vesicle picture, then the first step is fusion of the vesicle to the cell membrane. And that's because the electrical current charges each vesicle, and the charge, the electrical difference between the charge particle in the vesicle and the neutral of the synapse, that is the potential. That's the voltage drop. And so when the vesicle actually reaches and fuses to the cell membrane, that voltage impacts the calcium channels, which open the gate. And these are called G-coupled proteins or calcium channel gates. Interesting stuff, how it all works together on an electrochemical basis. And so the gates opened at 2, and at 2, we start seeing evidence, and the measurements at the top here at what's labeled C, the graphs, that's actually in the postsynaptic receptor. That's on the other side of the synapse. And you see that as neurotransmitter molecules, dopamine again, are released into the synaptic cleft, the receptors on the other side start activating, producing electrical charge, to a peak at 0.3, as shown in the graph. And this doesn't happen instantly. And when we go back to the very beginning, if you remember the Sapolsky lecture about how many synapses um, for each circuit in the brain, and that the fewer synapses, the faster the signal and the more dominant the signal. Say something 2 synapses away versus 10 synapses away, like instinctive reaction to someone poking you in the back with a sharp object, is 2 synapses away. It produces the instant recoil, self-defense, not thinking, and the longer path to prefrontal cortex that is going to sort out what's actually going on for real, how would be the socially appropriate way to act. That's 10 synapses away. And it's enough time delay. 2 synapses, 12 milliseconds. 10 synapses, 60. This matters. And so, as part of maintaining homeostasis, meaning constant amount over time of dopamine in the synapse, uh, in the synaptic cleft, in the presynaptic terminal, to activate receptors enough to keep them stimulated for staying alive, survival. Once neurotransmitters are released into the synaptic cleft, they're recycled back. The recycling process happens, and that achieves, one, a way for the neuron to actually control the levels within the synaptic cleft. I meaning, based on stimulation, control how much stimulation is being sent to the next neuron, super important. And the availability for the next major stimulus event. If you don't have, say, dopamine in the synapse, the next great thing that comes along is not going to produce reward. There just isn't enough dopamine to be released. And that's one of those horrible pieces about addiction in the early recovery days, is there's just a depletion of the dopamine vesicles. There's just less. And this is interesting and worth giving some thought to, although not entirely understood. But the best minds in neuromicrobiology talk about a kiss and run with, in terms of the vesicles. And what we're talking about here is vesicle circulation, recycling. And so the kiss and run says they touch, they fuse, release, and then drift back into the presynaptic terminal to refill with dopamine. That's the kiss and run hypothesis. And then the other is actually that they're constructed from proteins from new within the cell. that could potentially be coming from the cell body itself, or actually manufactured by the endoplasmic reticula within the presynaptic terminals. But that's not entirely understood what's happening one way or the other there. And this may, depending on the order that one takes the TED's advanced program, we also touch on this in the neuropharmacology. And just to know that there are two types of receptors. And it's interesting, not incredibly relevant, but worth knowing. There's basically fast receptors, and what they do is a neurotransmitter opens a gate in the cell membrane, allowing ions to flow through it. As the diagram says, it represents that, and the gates are made of proteins. And so when a receptor, they have spaces for docking for molecules such as dopamine to dock at, but actually they don't work. Dopamine doesn't work on this basis. But the major neurotransmitters in the brain, which are glutamate followed by GABA, excitatory inhibitory balance, these are fast receptor neurons. And so what happens is a GABA or glutamate bind to the protein on the receptor, channels opened, and electrical charge passes through. And referring back to pharmacology, or store this upstairs to remember this when I talk about benzodiazepines. Benzodiazepines, actually, the Valiums, alprazolams, that class of medications actually works on the fast ion channels, meaning they attach just as a GABA molecule does. They will attach to exactly the same place and open up the same channels, producing just like GABA is meant to, meaning inhibit, reduce stress. And so, if one wants to spend the next 6 months drilling down into the really complex looking situation called G protein coupled receptors, GPCRs, these are considered slow receptors, and it's a comparative. It's a 6 millisecond versus a 1. And slow receptors, this is where dopamine inspiring addictive substances work on these receptors, meaning that, okay, let's take cocaine. We know that there's an increase of dopamine in the synaptic cleft because reuptake is blocked. The reuptake transporter is blocked by cocaine, so there's lots more dopamine flowing around. Those dopamine molecules actually act on G protein coupled receptors. And G protein coupled receptors versus ion receptors are a little bit different, meaning there's at least a two-stage event that happens. But what happens essentially is that the neurotransmitter could be dopamine, could be an opioid hitting an opioid receptor. That actually, all that does is create enough energy to then open an adjacent ion channel. And how the metabolism from dopamine hitting receptor into producing energy can be a multi-step process. Results the same. It's a release of charge within the postsynaptic terminal, and action potential or electrical current is passed on. If you have a look at this, and I think this is so well studied, because I think we've discussed before that I believe anyways that this is known so incredibly well because A, it's so important, and B, if pharmacologically mean produce a drug that blocks addiction, prevents addiction, wow, this is way more than discovering Viagra by accident. And when there's a whole lot of processes involved in a chemical reaction such as this, that also offers a lot more sites for pharmaceuticals to act and block. So I think that's why this is so well known, because whether it's fast release ionophore channel, meaning Valium or GABA, or whether it's a slow channel, even the 1 millisecond versus 6 millisecond, the overall, for the person actually using drugs, the limiting thing is actually how fast the body absorbs and transfers it into the bloodstream. And so, just to summarize what I've talked about, and I'm hoping this will help in terms of the next discussions and understanding how drugs work for real. To summarize, fast-acting neurotransmitters, GABA, glutamate, these act on ionophore channels and directly influence electrical excitability across the membrane. They act in milliseconds quick. And point-to-point communication, general homeostasis of the brain, it's a living thing. This is all the ultra-fast stuff that's just keeping things going. In general, the slow-acting neurotransmitters acting on G-coupled receptors, giving the response to inputs, meaning environmental change, slower, act in seconds to minutes at times because of the complexity. And what they do is they control how the rest of the neurons are working. And where do most drugs of abuse work? Slow acting. Anyways, I hope you've got something from this lecture. Thank you.